Hi everyone. In this lecture, we'll continue with RNA processing in eukaryotes. And in the previous lecture, we have seen a part of the RNA processing, that is modifications done at the 5' end, capping, and modifications done at the 3' end, poly A tail. After adding the 5' cap and poly A tail at the 3' end, a part of the processing has been done. It's still pre-mRNA. So what further modifications are required for the conversion of this pre-mRNA or transformation of this pre-mRNA into mature mRNA? That we will see in detail in this lecture. The pre-mRNA or the primary transcript immediately after transcription in eukaryotes is called as premature messenger RNA. It's not the final and it cannot be translated. We have seen this in the previous lectures. But why it's not final? Why it can't be translated, the premature messenger RNA? Because it has introns. What are introns? Introns are the non-coding sequence. So in eukaryotes, in general, the protein coding genes, genes which code for proteins, typically have non-amino acid coding sequences called introns between the other sequences that are present in mRNA, the exons. So the primary transcript pre-mRNA has introns which are non-coding. When we say non-coding, they do not code for any amino acids during the translation and they have to be removed. And the other sequences are the exons, which are the coding sequence. Intron is derived from intervening sequence, a sequence that is not translated into an amino acid sequence. And exon is derived from express sequence or the coding sequence which can be translated. The exons include the 5' and 3' untranslated regions. We have done the structure of mRNA in one of the previous lectures. And exons include not just 5' and 3' UTRs, but also amino acid coding portions. In the processing of the pre-mRNA to the mature mRNA molecule, the introns are removed. Let's see this diagram. This is the DNA. Or the gene. And this is the promoter sequence. In the initial lectures, we have learned the significance of a promoter sequence, how the RNA polymerase enzyme orients itself to the promoter and understands which of the strand is the template strand, in which direction it has to move for transcription, and which is the first nucleotide to be copied, that is +1, that is where to begin the transcription. And since we are learning RNA processing in eukaryotes, eukaryotes have 3 RNA polymerases, and it's the RNA polymerase 2 which carries out transcription of the protein coding genes. And there are general transcription factors also involved. So transcription by RNA polymerase 2 results in this premature messenger RNA or pre-mRNA. There is addition of a 5' cap when only 20 to 30 nucleotides of the pre-mRNA are made. This cap is visible here. In the previous lecture, we have seen this cap is a guanosine nucleotide, which is attached via an unusual 5' to 5' linkage. And then that guanosine is methylated at the 7th position with sugars of the next nucleotides also getting methylated. And there is also addition of a poly A tail at the 3' end for which there is no DNA template. So this cap and tail addition is one half of the processing which we have seen in the previous lecture. In this premature messenger RNA, the cap and tail have been added. But there's one more interesting thing here. The two regions are shown in two different colors. The parrot green color thing are the exons, which are the coding sequences or the express sequence. And the peach color segment are the introns, which are the intervening or the non-coding sequences. These introns have to be removed and exons have to be joined. Pre-mRNAs have many introns, and these introns have to be excised. In the next step, it's evident that these introns are excised and the exons are joined. So, resulting in the production of mature mRNA. The peach color segment is not evident or not visible here, which means the introns have been excised or removed. And the three exons in the picture, exon 1, exon 2, and exon 3, have been joined into a continuous sequence. And the cap and the tail are still there. They remain throughout the processing. And this mature mRNA gets exported to the cytoplasm and translated into the polypeptide. Let's see more about this process. So, how the cell recognizes or the machinery recognizes where the introns begin and where the intron end? Because is there a possibility that accidentally an exon is removed, an intron is retained? That should not happen, and that doesn't happen because this machinery which does this is highly accurate. So exons are coding sequences. They have to be retained. So they are not removed. Introns are non-coding, only introns are removed. So what tells this machinery that does this, that where does an intron begin and where does an intron end? So introns begin typically with these two nucleotides, guanine and uracil, and end with these two nucleotides, adenine and guanine. So they begin with GU and they end with AG. There's a GU at the 5' and an AG at the 3' end. This is the 5' junction. This is the 3' junction. Let's say this is 5' junction and this is 3' junction. Although more than these nucleotides are needed to specify a junction between an intron and an exon. The process of conversion of this premature messenger RNA into mature mRNA is called as mRNA splicing. So if I say the process of conversion of pre-mRNA to mature mRNA is mRNA splicing, basically this process of removal of introns and joining of exons is called as mRNA splicing. And this mRNA splicing or RNA splicing takes place in the nucleus. So the transcription also takes place in the nucleus, and this RNA processing, that is RNA splicing, also takes place in the nucleus. Now we will come to the machinery that does this mRNA splicing, that removes the introns and joins the exons. This splicing events occur in a large macromolecular complex about the size of a ribosome called as spliceosome. So these splicing events occur in a macromolecular complex called spliceosome. What is spliceosome? Spliceosome, yes, it's a macromolecular complex, but this spliceosome is the complex of pre-messenger mRNA, obviously, which has to be processed, the target, and small nuclear ribonucleoprotein particles. Small nuclear ribonucleoprotein particles called as snRNPs. They are pronounced as SNRPs. So, what are we talking about? RNA processing in eukaryotes. Pre-mRNA has to be converted into mature mRNA. Yes. Pre-mRNA has non-coding sequences called introns, which have to be removed, and exons, the coding sequences, have to be joined. This process is called as mRNA splicing. These splicing events occur in a complex called a spliceosome, or the machinery which does this splicing, which removes the introns and joins the exons. And this spliceosome is a complex of pre-messenger RNA, the target, bound to SNRPs. What are SNRPs? Small, yes, they are small, nuclear. They are present in the nucleus, and the splicing takes place in the nucleus. Ribonucleoprotein particles. Ribonucleoprotein particles means, in simple words, a combination of RNA and protein. Combination of RNA and protein. And when I say small, the RNAs are small nuclear RNAs. The complex is large in its own, like about the size of a ribosome, as I said. So if these NERPs are combination of RNA and proteins, which RNA? So in one of the earlier lectures, we have seen that how the three RNA polymerases in eukaryotes carries out the transcription of genes coding for these four types of RNAs. So one was messenger RNA, which codes for protein. Second is transfer RNA that brings amino acids during translation. Third is ribosomal RNA that forms part of ribosome. And that time we had said we will talk about that later, snRNA, small nuclear RNA. And the small nuclear RNA, along with proteins, forms NERPs, small nuclear ribonucleoprotein particles, which removes the introns and joins the exons. So these SNRPs have large RNA protein complexes with many proteins, more than 100 proteins participate in this activity. And the small nuclear RNAs of SNRPs are associated with proteins. The small nuclear RNA recognize sequences on the primary transcript by base pairing. Since it's snRNA and RNA, it can form bases with messenger RNA or premature messenger RNA, and they recognize certain sequences on the primary transcript by base pairing. And as I said that introns begin with GU and end with AG, but this base pairing also occurs. The snRNA recognize introns. Some of the sRNA molecules recognize conserved sequences at the splice junctions and understand where exactly the intron begins and intron ends. Once the splice junctions at both ends of an intron have been located by the snRNA, the proteins along with the snRNA in the spliceosome complex cut out the intron from both ends and join the exons together. And what is the fate of the intron which has been cut and removed from the pre-mRNA? The intron is removed as a lariat structure that is degraded, releasing free nucleotides. We will end this lecture by summarizing the entire process with the help of this diagram, which is similar to what we have learned so far or the other diagrams we have seen. This is a eukaryotic cell. The shaded area is a nucleus. This is gene or DNA, and the DNA has exons and introns. There are two introns in this gene, or the part of the gene which is shown, and three exons. After transcription, this is the primary RNA transcript still in the nucleus. This is pre-mRNA. Cap is added, tail is added, part of the RNA processing done, addition of cap and tail, and introns are removed. Who removes the introns? Machinery. Which machinery? Spliceosome. What is spliceosome? A complex of this pre-mRNA and SNRPs, small nuclear ribonucleoprotein particles, which are combination of snRNA, small nuclear RNAs, and proteins. So the splice junctions have been recognized, complementary base pairing form, and once the snRNAs know where the intron begins and ends, the introns have been excised. exons are joined, 3 exons are joined into a continuous, and this mature mRNA is exported to the cytoplasm and translated into protein. Humans, animals, plant, genes have many introns, and it's important that they are recognized by these machineries and excised, and exons are joined. So this is mRNA splicing, and with the 5' and 3' modification done in the previous lecture and mRNA splicing together, these three things become part of the RNA processing in eukaryotes.